Greetings to all of you and welcome to Cota Access. Today we are going to talk about a novel gene editing platform referred to as CRISPR. This session is part of Cota Access, a recurring webinar that we host several times a year, where we bring together founders, thought leaders, and leading executives to explore a variety of business and technology trends and topics that fundamentally impact our society. Cota Capital is an SEC-registered multi-stage investment firm based in San Francisco, focused on US-based modern enterprise technology companies. Cota has invested in over 75 private and 25 public technology companies since its inception in 2015 in the fields of infrastructure, workflow automation and applications, fintech, and health tech. My name is Kian Beyzavi, and I am a venture partner at Cota Capital. It is my pleasure to introduce our illustrious panelists today. We'll start with Daniel Dornbusch, a dear former colleague and CEO of Excision, a private company that is developing CRISPR-based therapies to cure viral infectious disease. And Amina El-Bibani is our next wonderful speaker. She is a principal at Artis Ventures, focusing on early stage tech bio, and that is not a typo, it's not biotech reversed, it's intentional, tech bio investing. So welcome to both of you. As we look at nature, we see that life is a constant battle for all organisms, right? Everyone thinks we have it the hardest, but few have it worse than the bacteria. Granted, we don't spend a lot of time on the well-being of bacteria unless it relates to our microbiome. But the truth is that the battle between bacteria and viruses that attack them is brutal. Bacteria are grossly outnumbered by bacteriophages, or these viruses, because there are more of these viruses on the planet than any other species combined. So more than the bacteria themselves, more than humans, more than mosquitoes on a hot, humid day. So bacteria have developed adaptive immunity to survive these attacks over the past billion years. I guess they have had their own Operation Warp Speed, but maybe at a different rate.
Yeah.
The mechanism that they use is called CRISPR, that helps them detect viral DNA and destroy it. So we have effectively borrowed CRISPR from bacteria. I guess you could call it a royalty-free licensing model to cure a variety of diseases in humans. So Daniel, I'd like to start with you and ask you if you could please explain exactly how CRISPR works.
Oh, wow. Thanks, Kian. And first, thank you so much to you, Christina, Allison, Simone, and the team at Cota for inviting us today. Always fun to talk about CRISPR. But to jump in and try to do what so many of the now Nobel laureates, of course, with Jennifer Doudna and Michel Charpentier have done to try to explain CRISPR in another way. My simplified version of it is that it is a protein that, as you said, has been evolved in bacteria for millennia to deactivate viruses, phage. These plague viruses, the way they use it is they actually have these proteins, and when they're infected with a phage, they're able to essentially, like we are able to recognize a flu or a new bacteria from time to time, we have a learning. Theirs is a little bit more rudimentary. They're taking pieces of an old virus, and that then they can use their CRISPR proteins to then deactivate the next time they see the same virus. The news and why this has hit our, why has this hit the news and why is it winning Nobel Prizes is really the ability to harness and change those bacterial proteins to do what we want with them, which is to direct them to all sorts of different and novel approaches using the efficiency of those proteins, which has expanded and exploded into so many different areas. Is that how much into the biology would you like to get?
So that sounds really interesting. Can you talk about how it actually works?
Sure.
So what does the protein do? Where does it go? How does it know where to go?
So if you go look at the bacteria and the way they use the bacteria in the royal form scheme, the way bacteria use CRISPR is they do have specific proteins. And actually, it turns out if you look across different bacteria around the planet, whether it's at the ocean floors or at the highest peaks of mountains, they're using different types of CRISPR editors. So when we use the term CRISPR, it describes hundreds or thousands, really, or many tens of thousands of types of these proteins. So the way it works is that they use two different pieces of RNA. And if you remember from high school biology, DNA turns to RNA, turns to proteins, at least in eukaryotes like us. But these are using RNA templates to then bind to DNA because they can complement each other and bind. So the way bacteria use them is they're actually using a tracer RNA and a CRISPR-associated RNA, so two pieces of RNA. You can either use two. We've now adapted it. You can just use one guide RNA. And what it does is it finds the exact sequence. It's usually about 20 base pairs, so ATCG, 20 letters long, binds to a specific area in the gene of targeting. Then the CRISPR comes, finds that RNA template, and makes a cut. Actually, it's kind of an elegant piece of, you can actually Google it, and there's even videos of CRISPR acting and working. You can see it's not all that clear because they're pretty tiny, but it is pretty amazing. It looks, to me, it looks a bit like a baseball glove with a little piece to it. And when it gets activated and binds to the guide RNA, it sort of just moves and cuts the DNA. But from there, it gets a little bit more complicated because how you cut it and where you cut it and in which ways, and do you have sticky ends, then it just depends on which editor you're using and how you're approaching it. So lots of versions from there.
Excellent. And I know you have a great YouTube video that that explains how CRISPR works, and there you can actually see the images. So that's great, and I encourage everyone to Google that.
It should be on Excision. Shameless plug, but yes, if you go to excisionbio.com or excision.bio, you'll see a little video on our website of how we use CRISPR, which is a little bit of an adaptation. So applying to human infected viruses or viruses that affect humans, and how our version of this, which is really to use two editors and remove large sections of viral genome. So that's what you'll see on our website.
Excellent. And so just to be clear, CRISPR is not an extra crunchy type of potato chip of British origin. It actually stands for clustered, regularly interspaced, short palindromic repeats. And I'm not going to repeat that. Amina, I'm interested to hear from you. Why now? Why are we seeing all the flurry of activity working on CRISPR now?
Yeah, absolutely. And thank you as well for putting this on and for having me. Daniel and I have these conversations on a regular basis, so it's great to broaden that and include more folks. Why now? I mean, certainly the discovery of CRISPR itself is a big factor as to why now, but I think really there are a few different emerging technologies and trends that have led us up to this point. The first, as many are probably familiar with, is the rise of genomic sequencing and our outpacing of Moore's law to really get it to low enough cost to be able to sequence a lot of different organisms around the world. And within that as well is the development of metagenomics as well. So our ability to now sequence microbial cells pulled from anywhere in the environment without having to culture them in labs where previously you would lose a lot of the diversity of those microbes prior to sequencing them. So now we can capture the full diversity of what's kind of out in our environments and really do a lot of discovery in those worlds as well. The second trend I'll mention is really the rise of cloud infrastructure. So folks now don't need to invest heavy capital amounts into building out analytics within their labs, but any scientist really anywhere in the world can now harness the power of the cloud and run a lot of their analytics within that. So as we're generating a lot of data, we're now also democratizing the analysis of that data. And that really lends into the realm of synthetic biology, which is, bringing engineering principles into the life sciences world. And just as important as it is to engineer and code these new organisms, and CRISPR as well, is to develop high throughput assays and engineering platforms to be able to, in very high throughputs, understand the functionalities of these components and of these organisms, to understand how efficient are they, what is the functionality that's actually occurring as we tweak and engineer things. So I'd say it's a confluence of a few different things, but exciting to see all of those converged now.
Excellent. And the outputs are amazing. I mean, that was beautifully put. But so you went through engineering, discovery, cloud infrastructure, synthetic biology, all of those come together, and you can actually give a high school student some of these tools now, and they can start doing CRISPR editing in their garage if you're an astute student. It's really democratized a lot of how gene editing works. I'm sure, Kian, you'll get into the positives and negatives of that kind of power, but yeah.
I'm waiting for the day that I can order on Amazon do-it-yourself CRISPR kits to grow 3 inches taller, but I may have to wait.
So, that one, maybe not.
So we didn't actually properly introduce you. I would love to have you take us through your backgrounds and your career paths, and tell us how you got to the point where you are today. So Daniel, we'll start with you.
Okay. So I was, I don't know, recovering scientist. Genetics in English undergrad, was doing a lot of molecular biology work over summers and during years in different labs at UC Berkeley at the molecular biology department under Ron Krauss when he was leading the department there. But then moved over to the business side pretty quickly, where it seems like I could help a little bit more. So Masters of Public Health at Tufts Medical School, with a dual degree at Emerson College, an MBA at Harvard. I started working at Genzyme, business development, project management, corporate development, M&A, then to Genentech, doing commercial work on Rituxan in the hematology division, then off to Novartis for several years in business development, commercial development. Ran the West Coast and Latin America for the commercial division for the diagnostics, which was really the infectious disease unit at the time for Novartis, and then worked in several small companies before joining Excision about 2 years ago. A little more, 2.5.
Fascinating. Thank you. Amina, how about you?
Yeah, so similar to Daniel, my background's in genetics. I also came from the academic world, had done, really came from the R&D world, had done research, both in academic settings, studying things from stem cell work to the microbiome to DNA damage and repair, and then as well some time in industry at Novozymes. I got into venture back in 2014, and it was partly due to really having a passion to see a lot of this fantastic technology that was coming out of universities and being developed in labs, and unfortunately just being published. And so, really wanted to lend my efforts into helping those types of teams be able to translate their technologies and ultimately be able to help out and impact a lot of patients. So got into venture in 2014, spent the first few years of my career there at another early stage firm where I came in to start and build out the health and bio practice. And so was doing quite a bit of early stage at this intersection of tech and life sciences. And over the years, as built out a lot of the co-investor network, Artis was one of the fantastic groups that I absolutely loved working with. So I joined the team here back in 2019, and really our focus is early stage tech bio, as you said, Kian. It's not a typo, and really it's our way of saying in reverse to traditional biotech, where a lot of things are single asset plays. We're looking at tech first approaches to the space, whether that's in the realm of computational platforms or whether it's in the realm of biological platforms, synthetic biology. Oftentimes it's a marriage of those two worlds, and investing pretty heavily from pharma to clinic facing plays to even outside of human health in areas like food tech, novel materials, industrial biotech.
And maybe the relevant part here, Kian, to our background, because Amina and I's backgrounds sort of overlap in an interesting Venn diagram, where she really brings in the different pieces. She's really deep. A little advertisement for you, Amina. Just your knowledge and experience of the different pieces, whether it's as you mentioned, cloud infrastructure, synthetic biology, engineering, the genetics side of it. You've seen really a broad array of different companies, technologies, and innovations come together in these I'm just old. I've just been hanging out on the gene therapy side for a while, and being a genzyme in the 90s, when so many of those great scientists, not me, but getting to work with them I was the business side of it, with seeing where they were doing so many of the early clinical trials on, this is before AAV, so doing trials on adenovirus and lentivirus and liposomes. Now we call them lipid nanoparticles, and just these so many technologies have just been around for a long time. But as Amina says, it's the technology she works with that has brought these into real use now.
You're entirely too kind and too humble, Daniel. But honestly, it's really the founders and the management teams that are bringing a lot of these. We're just here to support as investors.
Excellent. Thank you both. Amazing backgrounds. And I guess I'll go next. I'm also a former scientist, although I think you can't really turn that off. So, so I think once a scientist, you always think that way, but I've kind of evolved and added more skills to my toolkit. And so I started my career in electrical engineering and applied physics, but I put myself through college working as a research assistant in biophysics. And so since then, it's been a couple of decades, my work has focused on applying emerging technologies to life sciences. And I think we have that in common, that we are trying to look at cutting edge, like Amina put it very nicely, leveraging data, leveraging cloud computing and analytics to make great impact on the diagnosis and delivery of care. So excellent. Thank you all for that. I wanted to go back to Daniel and drill deeper on how is CRISPR different than other gene editing platforms that we have had, for the past few decades. In what way is it a step forward?
To get to the heart of it, the efficiency, ease, reproducibility. We've been working with gene edit. The royal we of scientists and biotech companies working with gene editors for decades. Zinc fingers, tau effector nucleases, meganucleases. There's all sorts of different versions of and really efficient ways to cut genes in specific manners. It's just never been quite so easy, efficient, and reproducible as with CRISPR.
Excellent. So faster, cheaper, better.
In a lot of ways, yeah, and not to disparage those others, they can be when applied in the right ways. Sagabo with their zinc fingers has done some really elegant work over the years and decades. But CRISPR has really made so many different things possible in the gene editing realms. In the case of us, we're actually delivering in vivo, meaning right into patients, multiple guides to enact multiple cuts into a viral genome. That really was not possible until CRISPR.
So actually, let's talk a little bit more about that. What are some of the diseases that you are targeting as your first indications, applying this revolutionary platform?
Excision's a bit of a different company when it comes to gene editing. We are one of the only ones, maybe the only company, targeting in vivo viruses, meaning viruses that affect humans. So in some ways, you could say that we're actually using CRISPR in the way it had evolved in bacteria to target and deactivate viruses. That said, we're directing it at humans rather than bacteria, of course. And to do so, we're doing it in an in vivo way, going right at the source. In fact, if you look through all of the viral treatments developed over the last several hundred years for viral infectious diseases, it treats symptoms, maybe lessens or shortens the symptomatic effects of different viruses. But we decided, since the dawn of this company, is let's go right at the source. It's the proviral DNA that's integrated itself into human genomes. Let's extract it, remove it, excise it, and in order to potentially achieve the goals of having a cure. So that's how we approach it. Now, we can go into the details of what we do and why CRISPR has transformed and made that possible today, but that's how we approach it.
And what are the disease states you're going after?
Oh, apologies, of course. Starting with, so really we could think about almost any viral infection, but our pipeline starting with an HIV program, herpes simplex 1 and 2, hepatitis B, as well as JC virus, which causes a rare, often fatal brain disorder called PML.
Yeah, I mean, so HIV, 38 million people worldwide, but we got a lot of questions regarding HSV1, the herpes simplex, also known as oral herpes, that impacts a staggering 4 billion people worldwide. So many questions from the audience have come through asking what your plans are specifically around HSV1. So I wonder if you can comment on any publications or any trials.
Yeah, you'll see a lot of our publications have been in the HIV space, mostly because that's our most advanced program, but we do continue and have been now building. You'll see on a financing strategy, we're relatively early. We just closed our Series A 2 months ago. So we are scaling up. You'll see some hopefully press releases in the next couple of weeks on scaling the team, labs, collaborations in order to move these forward and faster. But to get to your point, we are at a stage where we can now do this in vitro, meaning in cell lines, we can go in and show data to remove herpes simplex from specific infected cell lines. We'll be moving into animals this year. Keep an eye out for some news for new collaborations to forge in the next couple of months to start this work in vivo. Unfortunately, it's a little early for us to start specifying when we'll have a product on the market. We're still quite a bit away from that. And one of the reasons is, at least in the HSV approach, we've got a couple of options perhaps that we're going to be experimenting with. The first is how to actually deliver this. Does it need to be an IV administration, which would be a sort of a one-time option? Could we do this as a topical, sort of a sub-Q? Show up at a doctor's office, have a potential sub-Q. We know, or do we actually, and this is what we'd like to do, it's probably going to be our Series B and a topic of that fundraise later this year. But one of the concepts that we're starting to work on is, can we actually harness the same approach herpes simplex uses to after it and treat it? Meaning, can we use the same type of topical administration, because that's how we get infected, to go in and essentially go right up the axons to the peripheral nervous system, to the trigeminal ganglia and the superior cervical ganglia, the centers of neural tissue in the PNS that get infected with HSV, and go after it that way. So it may sound like rocket science or hocus pocus, but what we're really thinking is there a way we can do a repeat topical administration of a gene therapeutic to treat HSV? We're going to be years away from a product, but we're going to start working.
Excellent. And while we're on that topic, we are getting a lot of questions from Kimberly and others asking about HSV2. So can you touch on that also?
Yeah, well, that one's relative, well, at least it's a shorter answer. The same guides we're using, so going after HSV1 and 2. So the way we go about this for viral genomes, and really the reason we're so quite a bit different from other CRISPR companies who tend to focus on rare genetic diseases, is they're targeting human DNA. Where we're really just targeting viral DNA that shouldn't be there in the first place. So going after human genes that are mutated has a potentially higher chance. Well, actually, we can show it in all sorts of different publications that going after human sequences does have a much higher chance of these off-target effects, potential side effects, than what we're working on. But it's a long way of saying, by targeting homologous sequences that are shared between HSV1 and 2, because there are, we can potentially go after both with a single therapeutic.
Okay, excellent. And I think there are a lot of people in the audience that are very interested in that topic. So a couple of weeks ago, we heard the news that FDA approved the very first CRISPR therapy for humans that was for treatment of genetic defects that cause sickle cell anemia. Amina, I'd like to turn it over to you and have you discuss the other disease states that CRISPR could be used for.
Yeah, absolutely. And I think the exciting aspect here is it can really touch, many different disease states and even much beyond human health altogether. I think it began really in these monogenic diseases like sickle cell, as you mentioned, Huntington's, but really we're seeing it evolve now into more complex diseases, and especially a lot happening in the cancer and immunotherapy world as well. But beyond even just human diseases, if we take a step back and look at the microbial world, there are a lot of applications there. And infectious disease, Daniel already touched on the viral approaches, but I think there's also many that are targeting bacterial approaches, and we're very excited about those. To highlight one in our portfolio called Locus Bio, they're essentially harnessing those bacteriophages, loading them up with CRISPR, and really going in and targeting precisely infectious bacterial strains, so to get around some of these issues around antibiotics that we've now been facing for a while, where bacteria are now out evolving these antibiotics that we've had on the market for decades. And so coming up with really a new mechanism to come in, shred the majority of the bacterial genome and kind of tackle them in that way. So I think we're very excited that CRISPR is actually delivering and addressing a lot of these unmet needs and will be unmet needs in the near future. I mean, CRISPR itself as a tool, I think is incredibly powerful for the study of, genes, gene interactions and gene networks. And, ultimately, as we understand and build those maps of how each gene is interacting within a pathway across a disease, we'll be better able to create those therapeutics, whether it's a therapeutic through CRISPR itself or through another modality. And even beyond human health, there are a lot of exciting applications across agriculture. So how do we edit, seeds or crops themselves to either be able to grow more efficiently, increase yields in certain environments, or even to be able to withstand things like storage and transportation conditions, as they're going from farm to shelf. We're also seeing a lot in the food tech realm. So, areas of how do you edit either meat cells to be able to culture and create them in the lab, or differentiate them from stem cells to meat cells, or even how do you edit plant cells to more mimic the characteristics of those meat cells to get to more sustainable food development processes. So suffice that to say, there's a lot of different exciting applications, and I think we'll see a lot more, and perhaps even ones that we didn't expect in the next few years.
Mm. Excellent. Yeah, I was excited to see the news that a company is trying to use CRISPR for ALS and as you mentioned, for CAR T, enhancement of CAR T. So that's all really promising. What about we are in the middle of a pandemic, and I think every single diagnostic company or biotech company has been impacted by COVID one way or another. So, Amina, can you, as we are talking about use cases, has CRISPR been used in either diagnosis or treatment of COVID in any way?
Yeah, absolutely. It's certainly been employed in the diagnostics realm to develop rapid tests to detect COVID. In academic and research settings, it has been researched to potentially develop newer therapeutics and treatments down the road. But with regards to the current vaccines that we have on the market today, it hasn't been used. That's a different, very exciting technology around mRNA. But to my knowledge, CRISPR hasn't been used in those current vaccines.
Mm-hmm. Excellent. But for diagnostics, we are using it. Very good.
Yeah, absolutely.
So, Daniel, I'm interested in hearing from you as the CEO of Excision. What does success look like for a CRISPR company, and what are the elements that you need to succeed?
So I would argue that success in any biotech company is bringing products to patients. I mean, that's what you see in our mission is to relieve burdens of disease for patients. So we are absolutely targeting the best type of product to help rid the world of different, or rid humanity of at least one viral infectious disease at a time. So that's how we define success: is can we ease human suffering and death and morbidity and mortality. Mm-hmm. Is that fair for your or do you want to get in a little bit deeper on?
No, I think that's great. And I'm also curious to know, like, what do you need to succeed? I may not touched on all the different pieces that you need, and clearly the data science that's required for getting the guide RNA just right and knowing where to go and what to do. So I'm curious to know, like, what are all the elements, if you can talk about the skill sets, capabilities, and different functions that you need to bring to the table to succeed. What
these are quite diverse. What a good question. How long do you have? So part of it is, yes, CRISPR is absolutely powerful at an efficient gene editing. Now, the challenges of making this a reproducible, effective and safe therapy for patients around the world is manifold. Delivery in any gene editing, CRISPR or no, always has been and probably will be for quite some time a challenge. The most popular in vivo use of delivery technologies today is using AAV, adeno-associated virus, which actually is a variant of a type of variant of flu viruses, but it doesn't cause the flu. But it is an efficient vehicle, and it's already been FDA approved in other indications for specific genetic disorders. So it's a well-known and well-manufactured approach, but it has challenges, of course. It's expensive. It requires storage at -80, just like a lot of the COVID vaccines. You can only deliver it once. There's a whole host of new technologies coming, so we can put delivery into one. We actually have a part of our R&D at Excision. We actually have a team dedicated to Vector 2.0. So that's how we deliver the programs. The next is going to be just the editors themselves. How can we define what are the right editors and what are the right guides for each program that we're working on? Each of those is an entirely complex team, because it's not only just the geneticists who can look at, the guides match, but it's everything behind it, between where are the on target and the off target in the bioinformatics side, but also in vitro, then in vivo, the modeling, the assessments, the on and off target assessments. The data looks good, but it is a quite complex area. And we get into really the delivery, the manufacturing of it. This is always a challenge, or has been a challenge in gene therapy. A lot of great advances, especially in the past 5 years, and what we're looking at in the next 5 years, that things should transform another order of magnitude. But it's not making a small molecule. It's not making a pill. This is much more complex. And then getting this on a global distribution phase. I mean, if there are small silver linings to the COVID pandemic, perhaps one of them is establishing a global cold chain, as we're seeing now. This was not so easy to be able to store something at -80 and deliver it across the world to places who didn't have those refrigerators or trucks that could handle these. So there's all sorts of different components that we're working on to bring these to patients.
Excellent. And how will it actually be administered? Like, do you use an IV? Do you use a syringe? Is it systemic? Is it local? How do you administer it?
So for our first program, for EBT-101, this will be a one-time IV infusion. So the idea, at least how we're preparing the clinical trial to start this year, would be patients would need to be well controlled on their antiretrovirals for at least a year. They would come in as long as they pass the screenings, which is essentially, the age ranges. They can't have too many comorbidities. They have to be well controlled on their antiretrovirals for a year. It's a pretty wide open entry criteria. And then how it would be delivered, we would take the vials out of the -80 freezer, put them into an IV or put them into a single-use vial, very much like you see in the COVID vaccines. Mm-hmm. Bring it over to a patient, put it into an IV bag, administer it over an hour. It would look like and feel like a regular IV. At least that's the goal. And probably we watch everybody with their COVID vaccines for 15 minutes. We probably watch them for an hour or two. As the clinical trial protocol designers are putting it together now, and then the patient goes home and we watch. For the chance of, in the case of HIV, we'll be keeping them on, or at least the first clinical trial, we'll be keeping them on their antiretrovirals for 3 months and then stopping antiretrovirals and looking to see, watch for relapse, when and if that might happen.
So and one thing I'll add to that note as well is that to emphasize Daniel's point on the delivery aspect, I mean, especially as the industry as a whole moves forward and we start getting into more and more systemic in vivo gene therapy, really those delivery vehicles are going to be key, and developing new ones are also going to be key. You can think of, if a company is using AAV, that can then become an exclusion criteria for a company developing a gene therapy in a completely different indication, but using a similar vehicle. And a patient with that with those two comorbidities may not be able to actually participate in that trial or receive that therapy down the road because of their immune response. So I think it's great to see Excision doing this, and I think the industry as a whole, it's very imperative to think around novel delivery vehicles, whether they're viral or antiviral, to get around some of the immune response, payload capacity challenges, and the like.
Yeah, you're exactly right. I mean, and it's part of what you asked a question. I may have smiled earlier, Kian, but what do you expect out of some of the programs? Of course, the success is going to be bringing these products to patients. However, there's all sorts of milestones along the way that we think can advance the field to bring not only our therapeutics, but hopefully others to market as curative diseases, and or advance the CRISPR field. And what I mean by that is, as we go forward in these clinical trials, we'll be learning a lot. Biodistribution, how this works, the efficiency of different CRISPR editors with every clinical trial and really every publication. That's some of the questions I see here. With every publication, the field is learning and improving as we go. So we really do see some substantial advances as we move through these that we're excited to share with the world. So one of the things we like and we prioritize at Excision is publishing. You'll see a lot of the publications on our website to hopefully advance the field in those ways. So
I know the guide RNA tells the protein where to cut on the DNA, but when you administer the IV, how do these vectors know where to go? Do they kind of find their way into the lymph node and then kick into action, or?
Excellent question. So it depends on what delivery vehicle or vector is often referred to. We're using AAV9 because in the case of HIV, we need to essentially get to any lymphoid tissue in the body, including into the brain. AAV9 has been shown at certain doses to actually cross the blood-brain barrier, and we can get measurable doses into the brain. We've seen that in our mouse studies, our primate studies, and others have seen that in their human clinical trials. So that's why we're using AAV9, but there's all sorts of versions. People are using AAV2 and 6 and 8, 9, and then lipid nanoparticles and liposomes, as Amina mentioned, non-viral carriers, peptide carriers. There's all sorts of clinical trials ongoing. Last check, one last conversation I had with the FDA, so we've got over 1,000 gene therapy trials coming through these days. So the FDA knows what they're doing these days. There's some great teams at the FDA who are very well informed. So I've been looking forward to seeing these really get accelerated through, partially because the vectors and the carriers know. Now, others, just the other side of it is if you're dealing with a disease that is targeted to certain tissues. For example, our HBV program, of course, Hep B is only located in the liver. We're looking at something that's called tropism. We're looking for something that's specific delivery to a specific organ. In this case, liver happens to be one of the easiest things, right? You deliver anything IV and it ends up in the liver for the most part. So it's actually one of the easiest target organs for a gene therapeutic. There's other challenges with hepatitis B. How do we get every cccDNA, and do we need to get every integrated copy? We can discuss that if you like, but really the targeting has, what the point is, targeting has multiple levels.
Excellent, thank you. So let's now talk about what can go wrong. So they say only the paranoid survive, and there is some paranoia around the use of CRISPR. We are getting questions from the audience. Mike just asked about the impact of CRISPR on biodiversity. So as we talk about agriculture, I would love for you to comment on that. Daniel, I know we have had conversations around undesirable effects and what those might be. So let's kind of discuss what we think all the watchouts are and how we can go about it. I should also mention in 2018, many of you know that a Chinese scientist actually gene edited early stage embryos, and he targeted the germline or reproductive cells to make these embryos immune to HIV. And what's interesting in that case is the edited genes, because they were done in the germline, would pass through generations. And so effectively, that's like editing the human race. So what are your thoughts on all of these? Who wants to start? That's like a...
You want to go, Amina?
Yeah, I'll jump in. That's a big question and a lot of questions stacked together. I think in terms of agriculture, if we focus on that point first, for better or worse, and unfortunately, we already have lost a lot of diversity in the types of crops that we saw, centuries ago, just through, natural screening and reproducing and breeding that we've done for many, many decades. So I think it's a very good question. And I think it's one that, folks, as they go into development, should be thoughtful around. As we're editing some of these seeds to perhaps, get to better yields in certain environments, what are the known or unknown effects that can then develop, downstream? I mean, if we take the concept of editing embryos, as you mentioned, Kian, it is a very interesting question. And as in the US, we've been spending a few years talking about, how do we get the health system to become more preventative rather than reactionary and curative? I think that question in the context of CRISPR is one that is incredibly complex. And if you look at, I think very relevant for Excision here, they're looking at curing HIV, and then you take Dr. He, who looked to actually prevent HIV in embryos. I think most people would be pretty ethically aligned in those questions around curing HIV, improving patients' lives, and a lot of the suffering that they're facing today. And if we look then to kind of becoming more preventative questions, I think the lines there are a little bit more blurred. So I think it is very key to have transparency and communication across all of the stakeholders, around drug development, whether it is patients, providers, government and regulatory bodies, even investors. I think, VCs as coming in and enabling a lot of these teams to get their development and their products out on market. We also need to think about our accountability around, what are the potential, positive or negative consequences that might ultimately come from some of these technologies.
Thank you.
So much as you said, Amina, so many things to address here. So part of that Dr. He Chinese, there's a reason he's in prison. That I think the global community, from everything I've heard and everyone I've ever spoken to about editing germline cells or changing human genomes that can be passed on generation to generation is a red line. That we're just not doing that. I don't know of anyone doing that in humans. And if they do, they go to jail. It's a very simple piece. But Amina gets to the broader, deeper concept of, I think we can probably, at least as a community, say that the right application of gene editing is probably to alleviate diseases. How we go about that and make sure that we are not having unintended unintended consequences. We often talk about those as off-target effects. There's now great tools, as Amina can tell us better than I can, but there's so many great tools to assess this, to make sure that we're editing in the right ways and the right strategy. So we often target, to make it more specific, we often talk about 4 different places, and they're large buckets. So there's intended effects, so intended on-target effects, intended off-target effects, unintended off-target effects and unintended on-target effects. So the way we sort of think about these is when you go in and you look at, just think about one human cell, when you apply a gene editing technology product to it, where are their effects? Is it exactly only in the place that you are specifically telling it to edit? Or is there somewhere else in the genome that gets a cut? It's a key piece. So one of the things the FDA is, of course, most attuned to, are you making a cut in any oncogene? Is there any gene that could possibly create cancer when it's cut? And what are the odds you will cut it there? And they want to see quite a few cells, multiple cell lines, multiple tissues, multiple animals. Are you seeing anything? And so one of the challenges that we've seen in editing, take a look at whether it's Down syndrome or sickle cell disease, or take your pick on a rare genetic disorder, the human genome isn't random. We see repeat sequences throughout our genome. Yes, it's 3 billion base pairs, but there are repeat sequences that look very similar. So for example, if you're looking at the hemoglobin gene, you're going to edit a correct version of hemoglobin in for sickle cell disease, or clotting factors for any version of those genetic disorders. There are similar sequences throughout the human genome. So the odds of an off-target effect are relatively higher because there are similar sequences. We're in a space where we're, as I mentioned, only targeting viral sequences. So we actually can't even use the same software that other people use to assess off-target effects. Those tend to be only, and then you get into the bioinformatics of it, but to get a little bit more specific, I mentioned there's about 20 base pairs that you're looking at for those guide sequences. Well, this is biology. If you actually find one of those base pairs that's changed, for example, one of them is an A to a T or an A to a C, there is a relative chance of an off-target binding event, meaning even though there's one mismatch, it may bind somewhere else. If there's 2 mismatches, it's a lower chance. If there's 3 mismatches, it's even lower. In fact, once you get to 3 and 4, the odds are almost infinitesimal. We just don't see it biologically. But with 1 and 2 off mismatches, you can see off-target binding. So that's really where the bioinformaticians and the software programs, machine learning, we're applying so many different technologies to this to make sure that we're not going to have these off-target effects. It plays right to Excision's strength, which is we're not targeting human DNA. The viral sequences we're seeing in the retroviral genes that we target, in many instances, we're 4 and 5 mismatches away from anything we see in a human genome. That's a sort of long-winded way of saying we're trying to be very careful about going through this, and the regulatory agencies are wise to it and making sure that there's safety factors worked in.
Mm-hmm, mm-hmm. Very interesting. And, news came out from the Whitehead Institute at MIT and UCSF that scientists have now developed what they call CRISPR on, CRISPR off mechanisms using methylation to actually tag the portions of the DNA and suppress the expression of those genes. Do you have any thoughts on that in terms of how that could reduce off-target effects?
Yeah, you want to go, Mina, or you want to take it?
Go for it. I'll jump in.
Yeah, there have been lots of work going on with CRISPR and others editing, we call it epigenetics. Anything, if you're not actually editing the genome, are you editing somewhere downstream of that? So whether it's a methylation, whether it's tagging, whether it's adding specific moieties in order to apoptose, meaning kill a cell. There's been lots of work. I haven't seen any clinical trials off the top of my head using that approach, but it can be a really powerful way. So, for example, if you are not cutting DNA, you can actually skip past a bit of the off-target question. Right? If there are no cuts, you won't be looking at what were the chances of, and do we see any off-target effects? You would be looking at something slightly different. Are there any off-target methylation effects, and what are those downstream effects? I guess for those who aren't familiar with it, the idea being as DNA in the nucleus of a cell is translated to RNA and then to proteins, if you make certain adjustments, you can actually turn on and off genes, whether you methylate it or not. And the idea being, well, if you don't want something to be there, you can turn it off. And if you want more of it, you can turn it on. Then we get into the question of, well, what is the right level, and are you overproducing? But there's a whole set of different questions for each approach. But it's exciting. Sorry, go ahead.
Absolutely. I think it's very exciting, especially in the realms of R&D, if you're not having to go in and edit and cut out, genes in certain strains, you can then get to quicker iteration, turn off a gene, study it, turn it back on, turn off another gene and study what are the effects there. But to Daniel's point, I think off-target effects are going to have to be defined in a slightly different way with a technology like this.
Mm-hmm.
Which also leads you probably, Kian, I'm guessing your next question is, how can you do transient gene changes, right?
There you go. Let's talk about that too.
So there's all sorts of different ideas. You come up with an idea. I mean, this is maybe the power of CRISPR, is that now that it's relatively easy, you can have so many different versions of it. And so other people have said, well, how about if we just want to turn on a gene for a day, a week, a month, a year? Or what happens if we want to stop? So there's all sorts of different approaches, whether it is some people are using light-based, maybe you're actually going to use a certain type of, whether it's a UV or some type of light to activate the CRISPR. A lot of this work is being done in cancer when you actually want to make certain cuts in very specific cells and not everywhere else. Can you have an activating therapeutic? Meaning you give someone a gene therapy, it doesn't do anything until you actually add a chemical, whether you deliver that right into the tumor and then you only get gene editing right where you deliver that chemical, or whether it binds specifically to cancer cells and then actually turns on and brings in the gene therapy to cut those cells. There's some really clever ways that we'll probably be seeing in the next 5 years that are in clinical trials.
So just looking at questions, Luis just asked, how generic can these treatments be? So do you need to have a specific treatment per mutation of the virus, or do you do one treatment for the virus family?
Right, fantastic question. The answer is yes and yes. It depends on, so we approach this with a, and we can get to Amina in a second. You can tell us how we do this, Amina, in more detail. But we use quite a few software approaches and global DNA databases of viral sequences to select the most common sequences within the virus across populations. Meaning, nothing in biology is 100%, but we hope to target as close to, into the 90s % of all versions of at least HIV-1 to start. And we're looking, what we can do is just look at all of the sequences that have been discovered and cataloged over decades of the sequences of people's HIV. And maybe not surprisingly, some of the genes that are essential to HIV formation and replication are the ones that are actually shared across populations, right? If they're key genes, those are the ones that are probably not going to get mutated and be essential for HIV's activity. So those are actually the ones, if you remember your biology, we're targeting the LTR and the GAG regions of the HIV genome, two of the most specific and shared across populations. Those are the ones we're selecting. We've selected target sites that are shared as much as possible across different populations. And so the answer is yes and yes, that we're trying to come as much as we can have a one size fits all. But we know that again, this is biology and one size does not fit all. So there will be room for us to have ideally a version 1. And then if there's, for the populations that aren't treated with the version 1, version 2. Now, the nice part is that we can actually sequence people's HIV genome. So one of the entry criteria for our clinical trials is actually to come and take a look at do a blood sample, sequence their genomes, look for what version of the HIV sequence they have. Is there amenable to our guides? And if it is, then treat them. So a bit of a personalized medicine approach given the genomic tests.
Excellent.
Yeah.
And any comments, Amina?
No, I think that summed it up very well. I think, importantly, the big question is, not to find ourselves where we are with antibiotics today, and will, these genomes kind of out evolve a lot of the CRISPR therapeutics that are in development. But yeah, the key piece here that Daniel touched on is we can now sequence, so we can actually understand where the mutations are occurring and how is it out evolving, and then, take steps to make sure we're one step ahead.
Yeah, thank you. And in the last 3 minutes, I think no discussion of a therapeutic platform would be complete without touching on the question of cost. So, the science is great. We have shown clinical benefit, but at the end of the day, you want to enable access for a large number of patients that are suffering from a disease. We recently saw that the UK National Institute of Health and Care Excellence, NICE, rejected a novel gene therapy platform because of the price tag. So I'm curious to know what you all think about enabling access. And Daniel, I know you have thought about this for Excision. So if you could comment, both of you, would be great.
Want to go first, Amita?
Go for it, Daniel.
I'm just thinking about where to start. Yeah, currently gene therapeutics, those on the market are quite expensive, in the millions of dollars range. Now, those on the markets are also focused on very small populations with devastating diseases, often affecting kids and having terminal effect. Not the therapeutics, but the diseases are usually terminal in children. So if there are only a couple hundred, maybe a few thousand patients worldwide, understandable that in order to recoup the cost just to make the product, it may take quite a price to help to pay for it. We're targeting much larger markets. Obviously, as you mentioned, 38 million people worldwide with HIV. When you get to something like a hepatitis B that we're targeting, you're looking at 300 million, 350. We're seeing some numbers above that these days. So just those infected. Get to herpes virus, and it's even higher. So the ability, the way we see it, is there's several components. One is technological advances. There's several innovations that we're seeing in manufacturers that we're working with that have been able to bring down, at least at small scale today, bring down the costs orders of magnitude from where they are today. So fast forward 5 years when we hope to be on the market, that we could actually bring down the cost per dose quite substantially. The second is scale. As these roll out, we've seen with every other product on the planet, as products become manufactured at scale, like of computers when they were the size of rooms to the laptops on your desk, the cost of manufacturing goes down. And this is the components, this is the pieces, these are the expertise, and it's also technology in order to manufacture these. So as we try to scale up, and we see this with monoclonal antibodies, right? These were extremely expensive 20 years ago, less so today. It's never going to be as cheap as, in my opinion. I would love to be wrong. I don't think it's going to be as cheap as a pill, as in pennies per day, but we'd love to see this in ways that would be able to be rolled out not only to developed countries, but to developing countries in similar ways to other therapeutics around the world.
Yeah. So, and a couple other things, and we are out of time, so we don't have time to actually go into the answers, but something to leave you with. Audience are asking if nonprofit organizations can potentially support the human clinical trials. So with that, I want to thank you both very much and thank the audience for joining us. I think we want to end on hope and optimism that as we watch this platform evolve, we need to be more prudent and thoughtful about how we harness its potential. So thank you all very much. And I'm going to end with showing our legal disclaimer.